how's it going, you guys? So in this video, I wanted to discuss the ChiSquare test. I know that as we get closer to the AP exam, I'm getting a lot of questions related to the ChiSquare test, how to use it, when to use it. So all of that stuff will be covered in this video. Now, a few things that I want to point out. For this particular video, we're going to discuss the goodness of fit ChiSquare test. There are other types of ChiSquare test, but generally speaking, the idea is the same, and the example that I will use in this video is a goodness of fit ChiSquare test. You don't need to know too much about that, but I wanted to just mention that for other people who are watching this video. Also, when do we use the ChiSquare test? Well, I can answer that right away. We use it whenever we have some observed value that is different than our expected value, and we need to make sure that they are not significantly different from one another. So as you know, biology is kind of messy. You don't always get the expected outcome. So for example, if you're expecting 50% of the offspring of two individuals to be something, and maybe it's not quite 50%, maybe it's a little off, there's an element of chance in biology. So oftentimes we use the ChiSquare test to see is the observed value significantly different than the expected value that we were anticipating. Now, with any ChiSquare test, you will have a null and alternative hypothesis. The null hypothesis will state that there is not going to be a significant difference between the observed and expected values, whereas the alternative hypothesis says that there is a significant difference between the two and that there is something else at play there that we didn't or perhaps didn't anticipate or factor in beforehand. So it makes more sense. This stuff might be a little bit confusing, but it will make more sense if we do an example. So let me take you over here. Now I do want to put a little disclaimer. You do need a certain amount of biology knowledge, specifically in regards to heredity. So you need to understand Mendels principles and stuff like that to understand this particular ChiSquare test. So now, with that being said, for our example, we're going to take Mendels Cross. Now, if you remember, Gregor Mendel, he did a cross with pea plants. And one of his most famous crosses was the cross that he did with two different strains of pea plants that had two different color seeds, two different color peas themselves. So some seeds were yellow and some were green. He found out that the yellow seed trait was dominant over the green seed trait. And we talk about alleles being versions of a gene. So with this particular trait, there was only one gene with two different versions. There was the yellow seed allele, and there was the green seed allele. And in any organism, you have two alleles, if they're a diploid organism like the pea plants in this example. So during his P generation F1 and F2 generation crosses, he crossed two F1 generation plants. So he crossed two heterozygous individuals, hetero meaning different. So that this means that they have two alleles that are different from one another. So each organism has a yellow allele, yellow seed allele, and a green seed allele. Now because the yellow seed allele is dominant over the green seed allele, both of these individuals are yellow phenotypically, but they carry a green seed allele. Now if we were to cross them, you can see that I did a Punnett square. I already filled that out. But if we were to cross two heterozygous pea plants for this particular trait, we see that 3 out of 4 of the outcomes will give us a yellow seed phenotype, whereas 1 out of 4 is going to give us a green seed phenotype. So we should expect to see 75% of the individuals showing yellow seeds, and about 25% of the individuals showing green seeds, right? Now, you can see over here, I kind of listed this out. So out of our genotypes is the actual genetic makeup. That's the combination of alleles that we have. For the offspring, we have 25% of the offspring having a homozygous dominant genotype, we have 50% of the offspring having a heterozygous genotype, and 25% of the offspring having a homozygous recessive genotype. So that's genotypically the distribution, but the phenotypic distribution is 75% yellow seeds and 25% green seeds in the offspring. So these, we kind of use this Punnett square to generate our expected values. So hopefully that made sense. Now I want you to imagine that we crossed these two heterozygous individuals and we counted a total of 1,000 offspring, right? Now, if you were to do that, if you were to count 1,000 offspring with these two crosses, or with this particular cross, you would expect 75% of the offspring to show yellow seeds, and then 25% of the offspring to show green seeds, as displayed by the Punnett square. So, with 1,000 offspring total, you would expect 750 of them to have yellow seeds, and 250 to have green seeds. Now let's say we did this cross, and we didn't get these numbers exactly. We got 739 yellow-seeded plants and 261 green-seeded plants. So now you might be thinking, well, that makes sense. This is, there's an element of chance involved. We're not going to see exactly 750 yellow. Maybe 739, that's pretty close, right? Maybe we can just accept that. Well, in biology, if you want to do good science, especially when it comes to hereditary experiments like this, we want to be sure. We want to make sure that there's not some other element involved that we didn't anticipate, and that this number and this number are different purely due to chance and not something else that we didn't consider, if that makes sense. So this is when we would want to use the chi-square test. We want to make sure that this is not significantly different from this, and same thing with the green seed observed and expected values. So in this case, we do the chi-square test using the chi-square equation. So the chi-square equation, so this is the chi-square value. The chi-square equation is the sum, so this right here, this sigma, capital sigma, a Greek symbol, is sum, which means we're adding. So it's the sum of all of the observed minus their expected values squared over the expected value. So basically, we have the observed for each one, green and yellow. So let's just say we did yellow first. We would put the observed yellow seeds, which is 739, minus the expected, we expected 750 because it's 75% of the offspring. We square that. We square it because we want to get rid of any negative numbers, or we want to keep our numbers positive in this particular test, which is why whoever came up with this equation squared it. And then you divide that by the expected value. So in this case, if it's yellow, it's 750. So if we did this out, we would say 739 yellow seeds were our observed amount of yellow seeds, minus 750, which were our expected number of yellow seeds, seeded plants, square that, and then divide it by 750, which is our expected. So hopefully all of you can see that, but we're not done. We did yellow seeds, but we also have green seeds, right? So that's where the sum comes in. So we add this whole equation again, but now for green seeds. So it's 261 observed green seeded plants, minus our expected, which was 250. We square it. That would hypothetically get rid of the negative if there was a negative here. Divide it by 250, which is the expected number of green seeds. And then if we do all of this out, you don't necessarily have to show this intermediate step, but I did so you can kind of see the math. Our chi-square value ends up being 0.645. So this is our official chi-square value. I'll even write that in. Chi-square value. Now, a common mistake that students sometimes do is they actually square root both sides. You don't need to do that. So this right here is our final value. You don't need to square root both sides. This chi-squared is just that. That is the value that we're solving for. So now we have this value. Great. Now what? What use is 0.645 to us? Well, we can't just do the equation, or we can't just solve for the chi-square value. We have to compare it to something. And on the AP exam, you guys are going to get a table called the ChiSquare Table. It's going to be on your AP Bio Equations and Formulas worksheet, which I strongly recommend you bring to the AP exam, which is online this year. But you will need to compare this to that table, which we'll do in a second. But before we look at that table, I also want you guys to know how to calculate degrees of freedom. So that's another value that you need to calculate, and it's super easy to calculate. All you have to do is say the degrees of freedom is equal to the number of possible outcomes minus 1. So in this case, we could have gotten yellow seeds, or we could have gotten green seeds. So we have 2 possible outcomes. So we do degrees of freedom equals 2 minus 1, which in this case, our degrees of freedom is 1. If we were doing a cross that was incomplete dominance, for example, where you crossed a pink flower with a pink flower, and you could have gotten red offspring, pink offspring, and white offspring. Now you have 3 possible outcomes, so your degrees of freedom would be 2. So hopefully that makes sense. If you did a cross like the dihybrid cross that we did earlier in the year, where you're not just considering 1 trait, but 2, so round and like the shape of the seed as well as the color of the seed, that gives you 4 possible outcomes, so your degrees of freedom would be 3. So hopefully that makes sense. Now, so we have our degrees of freedom, we have our chi-square value, so we bring that over to our table. Now, I couldn't fit the entire table on this board. I'll attach a picture, or I'll edit in a picture of the chi-square table that you will see on the AP Biology Equations and Formulas worksheet. So you can reference that if you want to do other equations using this video for support. But here we have our incomplete table. This is enough of what we need for our purposes for this example. So here you see that there's we have up to 5 degrees of freedom, where it cuts off, and we have our p-value. Now, I'll explain what that is in a second, but we have a chi-square value. I'll rewrite it over here, so this way we can reference it. So our chi-square value that we solved was 0.645. So that was our chi-square value that we just solved for, and I'll put that up there for our reference. Now, we have that. How do we use this table and that equation to figure out whether or not our observed and our expected values are significantly different from each other? Well, we also calculated that we have 1 degree of freedom for this particular example. So we have 1 degree of freedom, and our chi-square value is 0.645. So these values right here are our critical values. We sometimes represent them symbolically using the chi-square symbol plus a little c, subscript c. So that's our critical value. Now we basically, for our degrees of freedom, which in this case is 1, we figure out which critical value is closest to our chi-square value that we calculated. Well, our chi-square value was 0.645. Take a second to look at this. Is 0.645 closer to 3.84, or is it closer to 6.63? I'll give you guys a second. You guys got it. So it is 3.84. 3.84 is closer to our value, our chi-square value. So our critical value in this case for 1 degree of freedom is 3.84. That will give us our p-value, which in this case is 0.05. Now 0.05 is a special number. Some of these chi-square tables will have large numbers of critical values that you can pick from. For the AP exam, they try to keep it simple with just 2 possible p-values with only this many critical values to pick from. But in this case, you get 0.645 for 1 degree of freedom. We have our critical value, which is 3.84. That gives us our p-value of 0.05. P in this case stands for probability, so it kind of says that there is a 5% probability that the difference is due to chance alone. And so if it's 0.05 or above for any chi-square test, then we go with our null hypothesis, which is the difference is due to chance alone. We basically, as they say in statistics, we fail to reject our null hypothesis, which for our purposes means that we accept our null hypothesis, which means that it is simply due to chance. So that those numbers that we got, our observed values versus our expected values, the difference between them in this case is just due to chance alone, because biology is messy. If our chi-square value was like 7.33, that's closer to this value, which means that our p-value would have been 0.01, which means that there's only a 1% chance that the difference is due to chance, which means we reject our null hypothesis and we accept our alternative hypothesis. The difference is not due to chance. That means that there was something else there that maybe we didn't anticipate. Maybe the 2 organisms that we crossed weren't actually heterozygous, but that's basically what we would have done if we got a value closer to this, a chi-square value closer to this than this. I have gotten in the past, what if we get a chi-square value that's somewhere in the middle of these 2? For the AP exam, don't worry about that. They're not going to give you anything like that, but let's just say, for the sake of argument's sake, that you got something like 3.9 or 4.0. In that case, you just pick whichever one is closer, which in this case is this critical value. But you wouldn't actually get that on an AP exam. It's either going to be below this, your chi-square value will either be below this or above this critical value. So hopefully that makes sense. Just to kind of summarize, just so you can see the big picture, our chi-square value is 0.645 for our values, our observed and expected values, which means our critical value is 3.84, which means our p-value is 0.05, and if our p-value is greater than or equal to 0.05, then we go with our null hypothesis. So we fail to reject our null hypothesis, and the difference is due to chance alone. So if we look at our values, we say that it's our null hypothesis, the difference is due to chance. So this difference between 739 and 261, and comparing that to our expected values of 750 and 250, this difference is just due to chance and not something else that we didn't factor in. So that just means that biology is messy, and it's very possible to get these values and not these values. This is close enough to this to say that our pattern of inheritance was correct, our simple dominance pattern. What we expected was pretty much correct. So hopefully that makes sense. I know that was a lot. I will attach another question like this to see, or for you guys to kind of see if you really understand this stuff, for you guys to test yourself. I'll attach that down below in the description of this video, but hopefully that was helpful, and I will see you in the next video.